Today, we are going to cover staph, strep, gram-positive rods, Neisseria, and Moraxella. This is basically just a study guide to kind of combine visual learning. I've prompted Gemini AI to create images. The funnier and sillier, the better, to kind of remember each of the bacteria and some of the important classifications. Um, Disclaimer, I am actually still a student. I'm making this and teaching it because it's a great way that I can learn for my classes for microbiology. So, don't take everything I say as truth. Certainly, you know, make your own, you do your own research, make sure everything's correct, but feel free to use this as a study guide for all the genus that I just described. All right. So you can see on the screen right now, we are starting with staph. And these kind of, workflows are great. I definitely recommend doing them on your own so that you can kind of recall them while you're in the test. You can work your way down gram stain positive, cocci, catalase positive, coagulase, and that splits the Staph aureus from the coagulase negative options over here. But we're going to go into a little bit more detail. This really isn't what the video is about. So we're going to move on to those kind of funny little images, graphics. Some of them Gemini AI did a great job at, and some of them are not so great. You'll see what I mean once we get down into it. Okay, so here's our little Staph aureus guy. Real quick though, let's do some general information about Staphylococcus. It is a gram-positive cocci in clusters. The colonies appear creamy, white, opaque, butyrous. They are catalase positive, non-motile, non-spore forming, facultative, anaerobic. They grow on blood agar, chocolate. They grow on CNA and PEA, which is selective for gram-positive. They do not grow on MacConkey because gram-positive growth is inhibited. And if you use MSA agar, it is selective for staph and it differentiates Staph aureus due to the mannitol fermentation, which turns it yellow. Some key tests, catalase, it differentiates staph, which is positive, from strep. Coagulase differentiates Staph aureus, which is positive, from all the coagative negative streps, and novobiocin, which will separate the coag negative species. So Staph aureus, again, gram-positive cluster, cocci in clusters, medium creamy, light gold yellow on blood agar. We want to see, so it is a positive coagulase test. This is the only Staph aureus, that is a positive coagulase test. It causes folliculitis. Looks like we've got folliculitis on there a few times. I don't even know what word this was trying to say here. I can't even see it in my notes, but it also caused food poisoning. It is, let's see, it's resistant against penicillin and methicillin, and the MRSA version of Staph aureus is. It is a normal cause of acne. It's a normal flora, and it can cause toxic shock syndrome from toxin 1, I think it is. Let's see here. Yeah. TSST1 for the cytokine storm. It is normal flora in the nose, especially in healthcare workers. It's very common to see that. You can see the little gold colonies there that's trying to show. And I think that's all we have for Staph aureus. Oh, and then that MSA, the mannitol fermentation, it's not shown in this image. Okay. Micrococcus. So these are large gram-positive cocci in tetrads. It is a yellow pigment on agar. I don't agitate plate. I think it meant agar. um, it is catalase positive, which I believe this cat's trying to show. Coagulase negative. Here we go, right over here. It is a modified oxidase or microdase positive. I don't believe I necessarily have that on here. It is normal skin flora, and it creates the red colonies on MSA. So it grows on MSA, but it does not change color like Staph aureus does. It's a pretty rare, actually, as a pathogen, but it can cause endocarditis. It looks like staph, but it is the yellow colonies. So here's the rarely causes endocarditis. Okay, next up, staph epi. So this is one of our coag negative, which micrococcus was also coag negative, but another one of those. And this one is novobiocin susceptible. All right. And so that actually, when you're working your way down, your testing, once you get to coag negative, next you're going to be doing novobiocin, and the staph epi is going to be on the susceptible side, and staph saprophyticus, which is what we're going to cover next, is the resistant novobiocin. So as epidermis, staph epi is normal skin flora as well as mucous membranes. It is the number one cause of hospital acquired UTIs, and that's typically due to catheterization. Or indwelling devices. So shunts could also be in there as well. And here's our little happy, or I don't really know if he's angry, but it looks like a cluster of eggs because it's gram positive. And we know that the gram stain positives turn purple with the crystal violet. Our little cocci are in clusters in little skin. So it's normal flora and skin. Here we go. Catalase positive, bacitracin resistant, and novobiocin susceptible. It also creates a biofilm, so that's what helps it crawl up those indwelling medical devices like the shunts and the catheters. Let's see, what else? Only coagulase negative staph and micrococci are routinely tested for full susceptibilities. All right, let's go ahead and move on to staph saprophyticus. So again, this one is novobiocin resistant, bacitracin resistant, coagulase negative. It's in that grouping of coag negative staph. It also causes UTI, but this is going to be the second most common cause. This is just kind of in like healthy young women or older males. It's not necessarily hospital related. It's normal skin flora and mucous membranes. I'm trying to think if there's anything else that we need to know about staph saprophyticus. Novobiocin resistant is the biggest thing because that separates it from staph epidermis. Staph epi is novobiocin susceptible. So that means that clear ring around the disc is formed, whereas staph saprophyticus, when you are growing it on the plate and you place the novobiocin disc down, the bacteria will grow all the way up to the disc. It is resistant. It doesn't cause non-growth. Here's just a little quick overview. You can't really see it all that great, so I'm not going to spend a whole lot of time on this slide, but all your staph, aureus, micrococcus, epi, and saprophyticus. Moving on to our strep. So this is my little general picture. It does not produce, or it doesn't produce the hydrogen peroxide bubbles when you do the catalase. So this really separates staph from strep when you're looking at gram-positive cocci, because they're both under that categorization. You're next going to do the catalase test. On catalase positive, you're looking at staph, and that's when the catalase will bubble like immediately. And if it does not bubble from the catalase test, it is going to be in the strep side, okay? Let's see if there's any other general features I should cover here. They are facultative anaerobes. Some need CO2. They grow on blood, agar, chocolate, CNA, PEA, not McConkey, because remember, McConkey is selective for gram-negative, and this is gram-positive, right? We are going to see a really big part of figuring out which is the bacteria we're looking at, is the hemolysis patterns on blood agar. We're going to talk more about that. These can also be categorized with their Lansfield grouping, and those are the cell wall antigens. So you have groups A, B, C, D, F, and G. And the key virulence factor for strep is that they have a capsule, especially in strep pneumoniae. Let's get into some of our streps here. Okay, strep pyogenes. So this is our group A strep. And remember, the grouping is the Lansfield group. They gram stain small translucent colonies, and they are beta hemolysis. So you can see here it attempted to say that, beta hemolysis. I think that's the plural. But essentially what it's trying to show here is that like no blood is left on the plate. It's a very clear, when you hold it up to the light, the blood agar basically just completely disappears because it completely lyses the cells. Let's see. It is PYR positive, bacitracin A disc susceptible, and the Lansfield group A. So let's see if we can find, okay, PYR positive, we spotted that. Bacitracin sensitive. So that means when we put the A disc down, so think group A disc for bacitracin, when we put that down on a colony that has grown, it will, or we're attempting to culture and grow, they will leave an open ring around it. It won't allow growth near it. Okay. The virulence factors, protein M, anti-phagocytic, and streptolysin, O and S, it's hemolysin. So again, highly, lots of hemolysis going on there. This one is never normal flora. So this is a true pathogen. It's not normal flora. We don't normally find it on the skin or in the body anywhere. It causes strep throat. It causes scarlet fever, FEV, AI, skin infections, rheumatic fever, and acute glomerulonephritis. So the kidneys, right? In serology, we can do an ASO titer to see if there's a recent infection. And we actually don't need to do any susceptibility testing with group A strep, because it is always susceptible to penicillins. Let me see if there's anything else we need to know. Like the skin infections, you got erysipelas, impetigo, necrotizing fasciitis. That sounds pretty horrible. And then post-infection, sequelae results in the rheumatic fever and the glomerulonephritis in the kidney. So that's kind of when you didn't get it treated fast enough when you had the strep throat and it's moved on to the sequelae. All right. Group B strep. All right, look at that happy little baby. These are also under the beta hemolysis. Clears the whole plate, right? Beta hemolytic. Wow, AI really did not do a great job there. It's bacitracin resistant. Let's see here. So that means it doesn't have the clearing. It allows growth when we put the A disc down. It is PYR positive. So that means it turns that little PYR disc pink. It's also heparate hydrolysis positive. Let's see what else. Oh, heparate hydrolysis. So it turns the solution purple. That's the positive on that test. Bacitracin resistant. It causes neonatal sepsis and meningitis. It's actually the number one cause. So not a super happy baby there. And it's normal flora in the vagina. So pregnant women around 35 to 37 weeks actually get screened using a PCR test, and they treat positives to make sure that they don't give it to the baby when it's born. Again, since it's beta hemolytic, it does not require any susceptibility testing because it is susceptible to all penicillins. All right. Okay, let's see. Strep pneumoniae. So didn't quite spell it right. Close enough though, right? These are the only ones. So it was important to note that these are lancet-shaped diplococci. So they are still gram-positive cocci, but they're the only ones in the strep group that are lancet-shaped diplococci. They are P-disc susceptible, optogen. They also are bile solubility positive. That's not in this image, but essentially you take liquid bile and you just put it on the plate and it'll completely wash away the colonies. And so what you'll see is, because this is alpha hemolytic, which causes a little green discoloration on our plate, you'll see the colonies haven't washed away, but you'll see underneath the color change still. So you got to look closely on there. That's the bile solubility. Their virulence is capsules, which is a major factor, which I think this little helmet is showing the capsule. And let's see, diseases. So it's a number one cause of a lot of things. So a pneumonia in adults, meningitis in adults, I think is this guy. So pneumonia, meningitis, and oh my goodness, this poor kid. The number one cause of otitis media in kids, so ear infection. And I believe this is showing that it is normal skin flora and mucous membrane flora. All right, viridans. Gram-positive cocci. It's in the alpha hemolytic group, so it causes green there. Green's it's not exactly a green splatter. It turns the red green on a plate. It's very, very normal flora in the skin, the GI, not really the lungs, because the lungs are sterile. So I think this might be showing upper respiratory. Typically, we don't see a lot of disease with it, but it is associated with basically something followed up of a dental procedure, an invasive dental procedure. So it can lead to endocarditis, especially with someone with damaged or artificial valves. And it can also lead to wound infections or brain abscess. Let's see here. It's bile esculin negative, so this is a little, nope. So it does not turn bile esculin black, which is the test we use on that. And it is optichin, so that P disc resistant. Okay. So when we're looking at, here we go, let's find it. Viridans are over here. So we've covered beta hemolytic, now we're to the alpha hemolytic. We dusted strep pneumo, and we're moving down and looking at the viridans group, and it's bile esculin negative. Okay. Where are we? Okay. This is Enterococcus. So we typically say like group D and Enterococcus. So the hemolysis pattern of Enterococcus can really be any of the above. It can be beta hemolytic, it can be alpha hemolytic, it can also be gamma or non-hemolytic. It is going to be bile esculin positive. So that's what this is showing here. It causes the bile esculin to turn black. It also is a positive on the 6.5% sodium chloride test. So here it is. What it does is it turns that yellow broth kind of cloudy. It's also PYR test positive. So that is going to turn your little white disc pink. Some of the diseases it causes is hospital-related UTIs, catheters, noscomial, yeah, hospital-related essentially. It can also cause endocarditis, so I think this is a little heart there. This is endocarditis maybe. Wound infections, bacteremia, so that's over here. I'm not sure what the disco ball is. Like it threw a party in this image and I'm not sure. And so the resistant version is VRE, and that is a major hospital problem. Okay. And I think lastly for strep, we're looking at strep galolyticus, and it used to be called strep bovis. I don't know why it added biotype 1. I actually didn't even ask it to add this part of it because no one calls it strep bovis anymore. But strep galolyticus, if we look at our little thing here, you'll see that it's group D, right? It's not listed here, but it's basically in all three of these. That means it can be beta, alpha, and non-hemolytic, which was the same for Enterococcus, right? Let's go back to that image. There we are. So with this one, it is going to be okay. Bile esculin positive. It's PYR negative. So don't listen to this. No growth. It's PYR doesn't grow anything. It's a test where we take our little white disc, and if you put the reagent on it, it doesn't turn pink. It's also going to be 6.5% sodium chloride negative, so it doesn't turn the yellow broth cloudy. You can't really see that there. Here's the our little gram-positive cocci. It is associated with endocarditis and colorectal cancer, so that kind of separates it from Enterococcus as well. But really, Enterococcus and Galolyticus, the PYR and the sodium tests, the 6.5%, are what separate the two of them. They're both bile esculin positive, and they're both can be beta, alpha, and non-hemolytic. Summary page. Feel free to screenshot that if it helps. Okay, we're moving on to gram-positive rods. Let's see here. So we're going to first be looking at our spore-forming, non-branching. So that is the bacillus. They are large aerobic gram-positive rods, spore-forming, survive long-term in the environment. Large colonies, often contaminants. Catalase positive, usually motile, usually beta hemolytic. The exception is Bacillus anthracis, which is non-motile, non-hemolytic. So Bacillus cereus, and I may be saying that wrong. It kind of reminds me of cereal too. So it is associated with food poisoning, fried rice syndrome. I think this is a little plate of fried rice. It is an enterotoxin. It is beta hemolytic, and it is motile. So I think these are the little like pili or flagella that are allowing it to move. Maybe this too. Oh, no. This is because it's a spore former. It can float around in the air. And this is probably beta hemolytic. Why there's a B there, maybe? That looks like a beta. Clinically, it is a self-limiting disease, the food poisoning. Bacillus anthracis. So this one can be a terroristic biochemical. I've lost my place. Here we go. Bioterrorism agent. But surprisingly, it's not really, it's where it's non-motile, non-hemolytic. That makes it sound like not quite as scary. Look at the little sheep in the background. It included this. So there's 3 different ways, like diseases that it can cause. One's cutaneous, one's inhalation, and one's in the GI tract. So the inhalation is called wool sorter's disease, flu-like, respiratory collapse. But the reason it's called that is because there can actually be enough Bacillus anthracis within sheep's wool that when someone is shaving them down and kind of like sorting through it, they can actually inhale enough of it to cause issues. GI, I don't really know. I guess it doesn't really show GI here. Ingestion is rare but fatal. Cutaneous, black escar. So that is something you need to know about that. I'm not sure what this is showing here. This is the medusa head. So the colony is gray, flat, irregular edges, medusa head appearance. That's cute. I'm not sure. A horse sorter. I'm not sure what that. Oh, wool sorter. I bet that is what that is. Yeah, he's wearing a wool sweater. Okay. I wonder what this thing is. Leave a comment if you know what no broken tire and red blood cells. Non-hemolytic. I think that's what that means. Okay. Nocardia. Okay. So this is again, we're at the gram-positive rods. It's a non-spore former and it's branching. It is found in soil. Here we go. Pulmonary if inhaled, cutaneous trauma abscess, actinomycotic myocytomas with sulfur granules. I think these are the sulfur granules. It is weakly acid fast. That really separates it from a lot of the other gram-positive rods. Let's see, chalky, crumbly, waxy colonies. I think that these are, it's also a slow grower. That's what the snail is here. Let's see, with nocardia, are we catalase positive? I don't have that one up. Hold on one second. Take us back to our image here. So nocardia is going to be catalase positive. That's why that cat is there. It is opportunistic and it infects in the immunocompromised. Okay. I was a little disappointed with the Listeria image here. I feel like they could have done so much more with the information I gave it. But anyways, Listeria, it is found dairy, deli meats, and what's interesting about it is that it can grow at 4 degrees Celsius. So it can grow in the refrigerator. Um, Listeriosis, sepsis, meningitis. So that's typically by eating it on the contaminated food. It's really, really important in during pregnancy. So it's not necessarily harmful for the pregnant mother, but it is devastating for the neonatal, the baby inside. It is 50% fatal with the baby. So that's why they tell pregnant women not to eat a whole lot of certain foods, including like deli meat, sushi, things that are known to harbor Listeria and allow it to grow in the refrigerator. Um, let's see. It is not a spore former. It's catalase positive. So there's no cat here. I wish they had done that. It's non-branching. It is motile and bile esculin positive. So again, don't see that here. But what it does do at 25 degrees Celsius, right there, it's motile and it does this umbrella pattern when we grow that out. And at 35 degrees Celsius, it is non-motile. All right, Cronibacterium. This is horrifying. It's normal skin flora. It's often a contaminant. In on the slides, you're going to see diphthroids, so club-shaped rods in Chinese letters, VLY palisades. I think this is the palisading effect here. So again, gram-positive rods. I think that's the positive. It's also catalase positive. I don't know why he's like screaming catalase positive while he's dying from his gray pseudomembrane, but he is. And there's a couple different species under it within the Cronibacterium that we need to know about. So catalase positive, non-motile, tiny gamma colonies on blood agar. So gamma means non-hemolytic. Okay, so we've got Chronidiphtheria. The diphtheria causes that gray pseudomembrane in the throat and basically can cause you not to breathe, suffocation. It's toxin mediated. There's also Coroni ulcerans, which is also diphtheria. Chronigium, device-related infections. So, maybe that's it. Here he is. Device-related. So anything medical devices going into the body. And urolyticum causes urethritis and urease positive. So that is inflammation of the urethra. And that's urolyticum. Urolyticum. We use methylene blue to see the metachromatic granules. Loeffler's medium enhances granules. Tinsdale agar, it has black halos for tellurite reduction. And then for toxin testing with diphtheria, we use the ELIK test. It actually is spelled ELEK, but ELIK, to remember. I don't know. PCR for the tox gene. And then prevention, we do have the DTaP vaccine, which is a vaccine for diphtheria. Okay. I don't even know if I should attempt to pronounce this. Erysipelothex. Okay, don't bash me. So the source is swine or fish, and typically we're going to see it in butchers, vets, or fishermen. It causes erysipeloids, which are red skin lesions. Erysipeloids. It's kind of in the name, so you should be able to remember that. It can also spread to bacteremia and endocarditis. So in the lab, it does not create bubbles because it's catalase negative when we do the catalase test. It's non-branching, but what really sets this one apart is that it is hydrogen sulfide positive. It's really the only one that's going to create that. Hydrogen positive in TSI, which is rare for gram-positive rods. Non-motile. See if there's anything else. I think that's it. Okay. Lactobacillus. Look at our little happy lactobacillus there. All right. It is normal vaginal flora. It creates lactic acid, which lowers pH, which is protective in the vagina. I think this is really funny. So a lot of time it is a contaminant in urine cultures. That's why you can kind of tell if someone doesn't have a clean catch if they see a lot of contaminant of lactobacillus because it's normal in the vagina. Long chains of gram-positive rods, tiny alpha colonies on blood agar. It is catalase negative, so it does not create bubbles when we do the catalase test. I don't know if hydrogen sulfide gas is stinky or not, but like it doesn't produce the gas. Like no tooting. Whatever, however you want to remember that. Let me make sure I'm saying that right. Hold on. Hydrogen sulfide. Okay. I just wanted to make sure I wasn't making an idiot out of myself there. Okay. We're almost there, guys. Bear with me. All right. Gardnerella vaginalis. Gardnerella, basically. This is also normal flora, but when we see overgrowth, like if we, for some reason, knocked out some of our lactobacillus colonies, it could be due to taking antibiotics or something else. It is associated with BV or bacterial vaginosis. The Gardnerella is. So overgrowth, Gardnerella is bacterial vaginosis. And it smells bad or a fishy odor. Basically, this is showing the whiff test where you add potassium hydroxide and it's going to give a fishy odor. And then they are also associated, like under the microscope, with clue cells. And these are epithelial cells covered in bacteria. Look at that bacteria just covering the epithelial cells. I think that's really all we need to know for Gardnerella. Let me make sure I said that right. Potassium hydroxide. Okay, moving on to Neisseria Moraxella. This is the last section. All right, we're almost there. I was really impressed with this image that Gemini gave me. Okay, so just some general characteristics that apply to Neisseria and Moraxella. They are gram-negative diplococci. They show flattened sides, the kidney bean or coffee bean shape, except for Neisseria elongata, which is a rod shape, and we'll see that here in a little bit. They are oxidase positive and catalase positive. They are, in terms of growth, they're fastidious and capnophilic, so they need carbon dioxide. You're going to see variable growth on blood agar. Really, really likes to grow on chocolate. There's our chocolate agar there, which we know that chocolate agar isn't actually chocolate, right? It's blood agar that's been heated, so the red blood cells have lysed, really allowing them to get all the yummy nutrients they need. And they're also kind of surprisingly on McConkey, you're not going to see any growth, which is surprising, like I said, because they're gram negative. They should grow, but they don't on McConkey. So Neisseria gonorrhea. Here we go. It is always a pathogen, and humans are the only host. So that means that gonorrhea is not seen as normal flora. It's transmitted by STI. I told AI to keep it PG when I said STI, as I think this is them being all cozy. He's missing a leg over here, he or she. So they've only got 3 legs between the two of them. Cool. Infection sites, I mean, what do you see with STIs? Genitals, rectum, pharynx, skin, eyes, just mucous membrane areas. Symptoms. So men are usually symptomatic, urethritis, pus. Women, on the other side, are often asymptomatic, and they don't typically catch it unless like their partner is symptomatic, or it kind of grows and develops into PID, pelvic inflammatory disease, infertility, and ectopic pregnancy. So it can kind of lead to that. Let's see. It's also the number one cause of childhood arthritis. Ophthalmia neonatorium, basically newborn eye infection. And what they do after a baby's born, it's prevented by using erythromycin eye drops. Key lab notes: gram stain, intracellular, extracellular, gram negative, diplococci inside PMNs. So the PMNs are white blood cells, leukocytes. Again, diagnostic in men, not in women. They are very, very fragile, so no refrigeration. Plate it immediately, and you don't even use a cotton swab. I think you have to use rayon because they can dry out. Yeah, use Dacron or rayon swabs. Cotton inhibits growth. Definitely want to do selective chocolate, MTM, vancomycin, colistin, nystatin, trimethoprim. That's kind of a specialized agar. And they eat glucose, only eats glucose. And I think it says like hot and steamy here because you don't want it to be refrigerated on the chocolate. All right, Neisseria meningitis. Another one I was really impressed with. It started giving me some really good ones. So virulence, it has pili and a capsule. Here's that little capsule there. It causes meningitis, meningococcemia, sepsis plus meningitis, Waterhouse Fredrickson syndrome, petechia, shock, DIC, rapid death. Spread in respiratory droplets, and it's common in close populations. So like dorms, which is what's being shown here, barracks, prisons. It's always stat. So you're going to do the CSF, gram stain, and culture, drop everything and do it because it is so deadly. Again, it's fragile, so no refrigeration because it'll cause drying. It really likes chocolate, selective chocolate, sites with normal flora, and it uses glucose and maltose. So think of meningitis, you add the maltose in addition to glucose. Okay. Neisseria lactimica. All right. This is normal flora in the nasopharynx, especially in kids. Carbohydrate use. So it does all 3, glucose, maltose, and lactose. So think of the L as all those. And it also includes L, G, and, there's no G. L, M. Anyways, it does all 3. I don't know why this person has hairy lips, but they do. This little image here is showing the normal flora, and then this Neisseria elongata. So this is the one that is a, hold on here really quick. This is a rod-shaped Neisseria. None of the other Neisseria are. Remember, I said at the beginning, Neisseria are going to be the gram-negative diplococci, And elongata is the only one gram-negative rod-shaped one. I'm different. Hi. Let's see what else we have to say about these. Moraxella, just saying it's normal flora. Basically everything up here: sinuses, ears, upper respiratory. Lactemica in the nose, nasopharynx. What else do I have to say about Moraxella? Yeah, normal flora, upper respiratory. It's an opportunistic pathogen. It can cause any of these things. So ear, sinuses, pneumonia. Large gram-negative diplococci, hockey puck colonies, so they slide across the agar. DNA ACE positive, asaccharidic, does not ferment any sugars, so it doesn't eat glucose, sucrose, or lactose. It grows on blood agar, chocolate. It's inhibited on MTM. Let's see what this last one is going to do. Yeah, so this is just kind of a little summary of a lot of these. Gonorrhea only eats glucose, likes the chocolate. Capsule for meningitis. It eats glucose and maltose. Neisseria lactemica. How is it related to kids again? Normal flora in the nasopharynx of kids. That's why there's a bunch of kids around it. But it ferments all of these, so glucose, maltose, and lactose. And Moraxella. Oh, no sugar for me. Poor little Moraxella. He or she does not ferment any sugars. It makes that hockey puck colony. It slides across the agar. And I'm not sure why he's got scissors, but he does. If you happen to know why he has scissors based on everything we've said here, let me know. Anyways, that's all for today. Thank you so much. This has really helped me learn, and I hope that it has helped you learn.